Welcome friends. So in previous lecture we have started milk chemistry and constituent. So in that we have covered definition and basic components of milk. Uh, then we have studied detailed component analysis of milk uh, and as well as various chemical composition of milk of different species. Then we have already we have studied the physical state of constituents of milk. Today we are going to study detailed component analysis or detailed study of constituents of milk. So as you know that milk is an important food for mankind and young ones of all mammals. As a liquid food, milk contains basically water, fat, proteins, lactose, vitamins as well as minerals. So unraveling the composition of milk and its constituent has been a challenging problem for scientists due to its complex nature. So these constituents are present in three phases that are true solution, then colloidal dispersion and fatty in oil type emulsion. So a detailed knowledge of composition of milk lead to the understanding of physicochemical nature of this complex biomolecule. It will also add to the knowledge needed for preparation and manufacture of milk and its product. So today we are going to study detailed component analysis. So first we will see milk proteins. So basically proteins in milk they are present in colloidal form or they are present in colloidal state. So there are two proteins principally found in milk. They are casein as well as whey protein. So in milk there are two types of proteins they are found. These are casein as well as whey proteins. So first we'll see casein. So casein is the major protein which contributes at 80% of protein. Okay. So casein is the major component of protein part of milk. So it is present in milk in colloidal state in the form of calcium caseinate phosphate complex. So basically casein present in milk in the form of colloidal state in the form of complex, and that complex is known as calcium caseinate phosphate complex. So the micellar particles of caseins are spherical. So basically casein exists in forms like micelles. So these micelles are spherical in shape and they exist in milk as a distinct particles. So each micelle of casein is bridged with various submicelles. Okay. So these micelles are bridged with or linked with each other with some specific bond. So the submicelles within casein micelles are held together by calcium phosphate by an ester linkage with serine hydroxyl group. Okay. So these micelles they are linked with each other or another micelles by calcium phosphate ester linkage with serine hydroxyl group. So the calcium phosphocaseinate are rather larger in size as seen by electron microscope ranging in between 30 to 300 millimicrons in diameters. Okay. So micelles they range in size from 30 to 300 millimicrons in diameter. So definition of casein. So casein may be defined as phosphoprotein obtained from raw milk at pH 4.6 at temperature 20 degrees Celsius. If basically casein is what? Phosphoprotein means what? It contains phosphate group. Okay. So casein is made up of amino acids forming a polymeric chain through peptide bond. You know all proteins they are made up of amino acids, and in protein or amino acids they are linked by peptide bond. Okay. So the linkage is established by reaction of amino group as well as carboxyl group. Okay. So if you want to isolate casein, so best suitable conditions are pH 4.6 and temperature at 20 degree Celsius. See, this is a picture that showing two amino acids they are linked by peptide bond. So carboxyl group and amino group they together joined by peptide bond to form dipeptide. In this way, these amino acids they link by using peptide bonds to form chains of amino acids, and these chains of amino acids further they form primary structure, secondary structure, tertiary structure. Okay, in this way the protein is formed. Then next, whey proteins. So lactalbumin and globulin fractions of whey proteins are the two proteins together which form about 1/8 of the total protein of milk and are described as soluble protein. So whey protein comprises basically lactalbumin as well as globulin, and these are soluble in proteins. Okay, so both albumin and globulin are soluble in milk in whey, but they are different in that while albumin is soluble in water, while globulin is insoluble in water but soluble in dilute salt solution. So you know that whey protein, these are basically comprises lactalbumin as well as globulin. So both albumin and globulin they are soluble in milk as well as whey. So but there is slight difference in that albumin is soluble in water, but globulin which is not soluble in water, but it shows solubility in dilute salt solution. So both are coagulated by heat. So okay, so you can coagulate this protein by providing heat, and besides heat means their coagulation depends upon different factors like holding temperature. So temperature and time of holding, so how much time you can hold on a specific temperature, then different salt concentration and pH of the solution. So coagulation of these proteins basically depends upon these three things, that is temperature and time of holding, then salt concentration and pH of solution. Okay. So whey proteins are the native proteins present in whey or serum and are constituent of whey. Okay. So whey proteins, they are the native proteins present in whey or serum, which is prepared after removal of casein through acidification. So how can we prepare serum? So you can just remove casein through acidification at pH 4.6 or by the action of rennet enzyme during preparation of cheese. So you can prepare whey just by removing casein. So beta lactoglobulin is the major whey protein of milk, and beta lactoglobulin basically it is globular in nature. It has molecular weight around 36,000 Dalton, which is an isoelectric point of 5.2. So it is heat labile protein. Heat labile means it is heat sensitive and get denatured on heating. Okay. So it is soluble in salt solution and it has high nutritive value. So it possesses free SH group in its structure through amino acid cysteine. So it basically contains what? Sulfur. Then alpha lactalbumin is the second most abundant whey protein after beta lactoglobulin. So this protein that is alpha lactoglobulin, basically it is present in human milk. It is present in larger amount in human milk. This protein is involved in biosynthesis of lactose. So lactose is the principal sugar in milk, and this particular protein that is alpha lactalbumin, it is involved in the synthesis of lactose. It has molecular weight around 16,000 Daltons with an isoelectric point of 4.2. So this protein alpha lactalbumin, it contains no phosphorus or carbohydrate. Okay, so this protein contains this protein does not contain any phosphorus or carbohydrate. So this protein is very rich in amino acid, tryptophan and sulfur. Okay, so this protein lactalbumin is very rich in this protein is very rich in tryptophan. So tryptophan is nothing but aromatic amino acid, and besides tryptophan it also contains sulfur containing amino acids like cysteine as well as cysteine. So due to presence of essential amino acids, this protein is nutritionally important in whey protein. Okay, so this particular alpha lactalbumin is very nutritious and due to presence of essential amino acids in this particular protein, so it is nutritionally very important as far as diet is concerned. Then next protein is immunoglobulin. So globulin of milk and blood, they are identical. You know, our blood contains different types of antibodies, and they are of class immunoglobulin, that is IgA, IgD, IgA, and these antibodies play a very important role in defending against various kinds of pathogen. Okay, so globulins of milk and globulins of blood, they are identical. So in colostrum and milk, they are both are present, but globulin in the form of immunoglobulin is found in much higher amount in colostrum. So basically immunoglobulin is mostly found in colostrum, okay, and milk, they are present in the they are present in both milk as well as colostrum. So in milk, globulin is present to extent only for 0.1%. So these globulins are, they are present in very trace amount. Okay, so while in colostrum it is in 6%. So in my previous lecture I told you that the colostrum, that is the first milk after giving to young one, it is very important. It is rich with different kinds of antibodies, and it provides immunity to newborn infants. So colostrum is very important as far as the infant's immunity and dietary perspectives are concerned. So the concentration is so high, and it causes coagulation of colostrum on heating. So basically immunoglobulins, they are mostly present in colostrum, and they provide immunity to infants. Okay, and after eating this immunoglobulin, they causes coagulation of colostrum. So this property of colostrum being lost as globulin declines the normal level of milk. Okay, so the both albumin and globulin protein possesses high nutritive values and supply all essential amino acids. Okay, so moreover albumin rectifies and low sulfur content of casein in the form of alpha lactalbumin and beta lactalbumin in normal milk. So as far as dietary point of view, so both these albumins and globulin proteins, they possesses a high nutritive value, and these both proteins supply all essential amino acids. So these are very much important as far as dietary point of view. So molecular weight of immunoglobulins is very high, ranging between almost 1,80,000 Dalton to 8 lakh Daltons. So traces of certain other proteins are also present in milk. So these include proteoseptoptone, then iron containing protein, that is lactoferrin, then flat globule membrane proteins, then lactolin, then free secretory component, folate, and vitamin B12 binding protein, along with various milk enzymes. So these are the proteins that are present besides principal milk proteins, and they includes proteoseptone, then iron containing protein, that is lactoferrin, then fat protein, then vitamin 12 binding proteins, and there are some proteins they are bind with milk enzymes. Okay, so this protein comprises around 30 in number. So almost milk contains 30 to 35 number of proteins. Okay, then non-protein nitrogenous substances. So besides protein, there are some components, they are not protein, but they contain nitrogen in their nitrogen as a component. So these are categorized under non-protein nitrogenous substances. So milk contains various nitrogen containing substances which are not in true sense protein. Their amount is 5% of total nitrogen found in milk, and these are popularly referred as non-protein nitrogenous substances or non-protein nitrogenous constituents of milk. So they are present in four forms such as amine, example ethanol, amine, then choline, then amino acids, these are the free amino acids such as lysine, leucine, etc. Then amino acid derivatives such as creatine, then creatinine, and other components such as morphine and milk oligosaccharides. So these non-protein component varies from season to season and has no biological value as that of protein. Then enzymes in milk. So enzymes, they are biocatalysts, and they are found in plants as well as animal cells, and enzymes are nothing but they all enzymes, they are made up of protein, and they alter the rate of reaction without altering themselves. That's why enzymes are known as biocatalysts. So they bring about most of complex chemical reaction, but they themselves do not enter into chemical change. So by using enzyme, what enzyme does, so enzyme accelerate the rate of reaction without altering itself. So they are colloidal and proteinaceous. So all enzymes are made up of protein, and mostly they are colloidal in nature, or they are present in colloidal form. So they are classified as per the chemical changes they bring about, such as hydrolysis, these are hydrolyzing enzymes, then oxidase, these are oxidizing enzymes, then reductase, these are reducing enzymes, and they are also classified on the type of substrate they attack. Example, proteases, these are protein breaking enzymes, then lipases, these are fat splitting enzymes, they mostly act on fat, then amylases, these are carbohydrate, these enzymes act on carbohydrates, and they break down different types of carbohydrates. Okay, so enzymes are susceptible to heat, light, and pH changes. So they are substances which enhance the activity of various enzymes and are known as coenzymes, and those which inhibit the activity are called anti-enzymes. Okay, so enzymes, they are very sensitive to heat, light, and changes in pH. So their activity mostly depend upon these factors, physical factors. Okay, so we'll see enzymes in detail in next lecture. Okay, so in that we'll be going to study different kinds of enzymes and their role they play in milk. Okay, so we'll study enzymes in detail in next lecture. Thank you.